TRBC estimation. TRBC estimation. For TRBC estimation, what is the dilution factor? Okay, dilution factor I said, not fluid. What is the dilution factor for TRBC? Yes, 1:200. Okay, that is dilution factor. Then what is the dilution fluid name?
Hames fluid.
Hames fluid. Very good. Hames fluid. Then where we will count the RBC? The Newborn chamber. In what area we are going to count the RBC? In the central square. So in the central square we are counting the RBC. Am I correct? Yes. Now, central square is subdivided into how many small squares? Actually it is 5 into 5, 5 into 5, and that is 25 squares. Okay. Out of these 25 squares, only 5 squares will be counted. What are the 5 squares? 4 corner squares and central square. 4 corner squares and 1 central square. We will count the RBC in the Newborn chamber. Is everything clear so far?
Yes, sir.
Then next one is what is the formula? Formula for counting TRBC?
Sir, n into 10.
Yes, n into
10,000.
10,000. Okay, so before discussing this n into 10,000, let us come back to the basics of newborn chamber. Tell me the depth of the newborn chamber, students.
0.1 millimeter.
Yes. Depth of the newborn chamber. Okay. Depth of the newborn chamber will be. I will show you a diagram properly. Right. So, see students. So this is the counting area. Okay. That if you see okay. Now there is a depression here. There are 2 depressions running parallelly. Then there is a elevation like this. Now I will show a simple diagram. There is an elevation like this on both the sides, and there is a drying system here. Drying. And here we are keeping a cover slip. Then counting the area, and this depth is 0.1 mm. The depth of the newborn chamber is 0.1 mm. Okay. Then yes. Then this is cover slip, everything done. Okay. Then coming to the newborn chamber, the large square has been divided into 3 into 3, total 9 squares. Okay. So the overall volume of the area of the square will be 3 cube. Okay, 3 millimeter cube, 3 into 3, and also depth is there, 0.1 depth. Okay. So each square is 1 mm cube. Okay, 1 mm cube is each square volume. Then this is 4 into 4 grid. You already know 4 into 4 grid, and this is 5 into 5 grid. This is 5 into 5 grid. Okay. Now the volume of a single square. Okay, the volume of a single large square will be 1 mm, and the volume of a single small square will be 0.05 mm. Okay. The volume of a single small square of the central square will be 0.05 mm. Okay. Yeah, these are the values of newborn chamber. Then what else we need to study here? I guess that's it. Then coming to the calculation. Okay, calculation of TRBC. So how you will calculate? Write on the calculation. Everyone, volume of area of RBC counted in 5 squares of central square. Volume of area of RBC counted in 5 squares of central squares is equal to 1/5 into 1/5 into 0.1 depth into 5, no, 5 into 5, then millimeter cube. Area will be calculated in cube. Okay, so millimeter cube. Yeah, that equal to 1 by 50 millimeter cube. That equal to therefore number of RBC in volume, in one volume, equal to 1 by 50 millimeter cube equal to N. Okay, N number of cells are present in this volume of in this area. Okay, then number of RBC in 1 millimeter cube equal to N into 50. That equal to 200. That means N into 200. Okay. N into 200. Then dimensions of RBC counted in 1 millimeter square, 1 millimeter cube per microliter will be N into 50 into 200. That equal to N into 10,000. So this 200 is the dilution factor. Okay, this 200 is dilution factor. So that is how you got N into 10,000 value. So at the end of the day, what we are doing, we are counting the cells in 5 squares. Okay, if the count is 290, 290 into 10,000. Whatever value you are getting, that will be the RBC count. Okay. So I just given derivation of how N into 10,000 came. Okay, we have included depth, volume, dilution factor, everything. Then we got N into 10,000. Clear, right? Yes. Now, how we are performing this TRBC estimation? There are 2 methods or 3 methods of TRBC estimation. Methods of TRBC estimation. So 3 methods are there. Visual method, okay, or manual method. Then other one is automatic method. Method. Now in visual method there are 2 types. One is microdilution method. Another one is macrodilution method. And automatic we already know. By series analysis we can get automatic methods. The significance of manual is we cannot rush automations, that's it. Then coming to the microdilution method, test is very simple. So there is a modified pipette, okay, with 0.5 grading at the bottom, and there is a diluting chamber, and there is an upper value that is 101. So this is RBC diluting pipette. Okay, what we will do? We will collect blood up to 0.5 mark. Okay, then we will collect up to 1.11 the diluting fluid. What is the diluting fluid here? That is Haynes fluid. We will collect Haynes fluid up to 1011. We will dilute them. Then we will charge this diluted sample in the Newbar chamber. Right? First you will clean the Newbar chamber. Then you will place the cover slip. After keeping the cover slip, you will charge the Newbar chamber from side. Right? So that's it. The problem with this microdilution is mouth pipetting. You need to do the mouth pipetting, and mouth pipetting is not safe, as we already know. So we came with the macrodilution method. Macrodilution method, write down this value, very simple. You will take a test tube. In that test tube, you are going to add 4 ml of Haynes fluid. In a test tube, you are adding 4 ml of Haynes fluid. Okay? Now from this 4 ml, you will remove 20 microliters of fluid. You will remove 20 microliters of fluid from this test tube. So the overall volume of the fluid will be 3.98 ml. Okay, because we removed 20 microliters. Okay. Now to this removed test tube, we are going to add 20 microliters of blood, or 1 drop of blood will be added to this test tube. You will dilute it, then you will take this sample for charging the Newbar chamber. Okay. So here we have avoided, we eliminated the mouth pipetting. Okay, we are using pipettes to do this one. Got it? Yeah. So you are going to perform microdilution only. Our Shudbir has modified the nice. Shudbir has modified the pipette. Shudbir. Okay, class listen. So this is the summary of TRBC estimation. Is everything clear so far? Okay. Then no. What is the dilution factor of TRBC? Dilution factor 1:200. Okay. What is the upper value of RBC pipette? 101. Okay. For WBC this will be 11. For WBC this will be 11. That is the only difference. And diluting fluid name?
Hence fluid.
Hence fluid. What is the final formula of TRBC?
N into
10 into
200.
10,000. N into 10,000. Okay. And where you will count the RBC?
The depth of animal.
In the 5 small squares of the central square. Right? Everything clear, right?
Sir, what is 11 and 101?
Yes?
So what is 11 and 101?
11, that is the achievement of dilution. They are the markings in these pipettes. 11 for TWBC, 101 for TRBC. That's it. Okay? Diluting graduations. Yes. Do one thing, students. Everyone open your WhatsApp group. Go to quiz and attempt the quiz right now. I will give first experiment marks after this class. Open the phones, everyone. Open your phones and answer the quizzes.